All right, the last bit of this unit then is the bit on respiration, and I'm going to probably get through most of the aerobic pathway this afternoon. If I don't, it doesn't matter too much, but it is quite nice to get it done in one go, because obviously, you know, it is a continuous process, so leaving it part way through doesn't really help. And you've got some typed notes in your handouts. They're slightly dated in the way that part of it is presented, but I will explain that. Some bits we need more than others, but they're just there, they're convenient, so you know, you can work from those, but I will be putting other bits on the board, and I'm going to work through it step by step on the board anyway. There's some introductory bits to do on ATP, and then an overview of something called respiratory quotient, which don't always put that in because it's not something that we particularly need. But the plan this afternoon is to look at the aerobic respiration that then continues on the pages after that. So you've got, and some of this should seem familiar right back from your first assignment on mitochondria. You've got glycolysis, Krebs cycle, and the electron transport system. You will never escape mitochondria ever, no. The anaerobic respiration bit we'll come back to on Wednesday, but we'll see how it goes. It is obviously a really complex subject, so you know, if you're not getting it, then let me know. But what I'm going to do is try and sort of make it make sense anyway. Okay, now if we were to start with the idea of respiration, can be a bit confusing because people have it in their mind that that's what happens in the lungs, and of course it's not. What happens in the lungs is gaseous exchange or ventilation or breathing, if you like. That's not respiration. Respiration is what goes on in the cells. Essentially, in terms of chemistry, it's a bit like a combustion reaction. Well, it's exactly like a combustion reaction because it's taking organic compounds, combining them with oxygen, and getting carbon dioxide and water out at the end. But it's a much more controlled process than combustion. Combustion wouldn't be an awful lot of good, would it? You know, if your cells took on fuel molecules and then just burned them, you'd get a lot of heat in little bursts, but yeah, other than that, it's not going to be particularly useful. So this is where we meet ATP. Now we have met it at various points as we've gone through other processes. I think when we talked about active transport and various other points, we keep mentioning it, but you'll see in the first section of the notes in your handouts, there is a little bit about it. So it's an interesting point to consider it, actually, because ATP, all right, it stands for adenosine, and then the TP is triphosphate. That's it. Now, the first part of that, the adenosine bit, consists of something that we've only recently met, and that's adenine, which is one of the bases in nucleic acids, isn't it? DNA and RNA. The A is adenine. Now, adenosine is adenine and ribose, which is the same sugar as you find in RNA. So actually, this is part of the same thing. If you think about what we did in the nucleic acids, we had nucleotides, yeah? Nucleotides were sugar, phosphate, and base. What we've got here is sugar and the base forming adenosine, but actually we've got 3 phosphates instead of just 1. So it's one of those situations where, you know, you think you've heard of something in one place and then it turns up somewhere else. Well, yes, it is the same substance. It's not doing the same job, but, you know, we do use the same chemical compounds for all sorts of things. So you might, you know, when we drew it for the nucleic acids, of course, we had our ribose, I think we drew probably like that, didn't we? All right, and then we put our phosphate on there, and we put our base like that. So that was, I'm not sure we drew it with the O in, but that was a nucleotide. That was, if it was A here, adenine, then that was a nucleotide containing adenine. But the adenine and the ribose, all right, the little block here in the pentagon, is adenosine. So in fact, all we would need to do to make it into ATP would be that. All right, so we are talking about the same sort of materials, and we can recycle stuff and use it for all sorts of different things. So don't be surprised, if you do come across the same words used in different scenarios. So sugar, base, and phosphate would be a nucleotide. There is another word, I just crosses my mind to mention this while we're there. The adenosine bit, without any number of phosphates, all right, whether it's one making a nucleotide or 3 making ATP, if you just take adenine and the ribose joined together, that's described as a nucleoside with an S. All right, so adenosine, this is just an aside, really. So adenosine alone, all right, which is adenine plus ribose, is known as a nucleoside. Not a nuclear. What happened there? I tried to do an E and a D in one go. Nucleoside rather than a nucleotide. Obviously, there's a lot of different nucleosides. You could have, let's say, you could change the sugar. It doesn't have to be ribose. It could be deoxyribose. And it might not be adenine. It might be guanine. So you could have a nucleoside called deoxyguanosine, I suppose. I don't think you could call it. Yeah, that's how it'd have to work. Deoxyguanosine would be a nucleoside as well. So it's a nucleoside triphosphate, really. And I mention that because there are other compounds that are energy stores, if you like. And of course, that's what this is. This is a form of chemical energy which we can use for anything, really. Any processes that require energy, you'll usually find ATP involved. Another common one, I mentioned guanine because that is another common one, GTP. Guanosine triphosphate, phosphate, is another one. So yeah, just be prepared to meet them in different forms. But we're familiar with the idea that you build this probably from ADP with the addition of an inorganic phosphate. That means phosphate that's not attached to any carbon compounds. And you've got this reversible arrangement. I have missed out the energy, sorry. It doesn't really work like that, does it? But we can join the phosphate group onto ADP, and in doing so, we've got what's often described as an energy store. And you'll sometimes hear the bonds between, well, this bond here between adenosine monophosphate and the second phosphate, and this one here described as high energy bonds. But knowing what you know about bond energies, of course, you can see that those two are phosphate to phosphate bonds, whereas the first one between the sugar and the phosphate is a different sort of bond, isn't it? So, think back to what we've done. So there's more energy in those two bonds than anywhere else, and you can make them and break them. And so this is, a very important part of energy storage and energy use, or energy processing, really. Now, the purpose of respiration is to generate ATP. So during respiration, we're going to be doing this. And then, well, energy use is going to release the energy for whatever purpose is required. So it might be, say, for active transport or something like that, muscle contraction, all sorts of things. And the process of adding this phosphate group onto ADP to make ATP, the process is generally described as phosphorylation. All right, we'll be back to that. That's why I'm putting it in there. So we can phosphorylate ADP to make ATP, and we can break ATP back down again to release the energy. And of course, there are going to be enzymes involved in this, and it's usually ATPase. You know, it doesn't have any fancy sort of name here, but it should work either way. And we will find ATPase as an enzyme embedded in the surface of the cristae, in the mitochondria, all right, using, well, a build-up of protons to actually generate ATP. But that's a long way down the line yet. We may or may not get to that point this afternoon. That little bit at the beginning of the notes in your handouts gives you a quote of ATP occurs in living cells at concentrations between 0.5 and 2.5 milligrams per centimetre cubed. That's not a lot. And it's used up very quickly. I think, generally speaking, there's about enough ATP in a cell to last about 10 seconds. So if you stop the cell producing energy, you know, if you stop its mitochondrial function, you kill it basically, or you poison it, then it can cope for 10 seconds and then it runs out of ATP. All right, so, you know, if it can't make any more, that's it. So yeah, when we were talking about, was it cyanide? We were talking about being a poison, weren't we? Poisoning respiratory enzymes. So yeah, it should have its effect, sort of, you know, 10 seconds worth of ATP production, and then if you can't make any more, that's your lot. So yeah, if you have got the antidote, then you need to take it very quickly. Right, this idea of ATP then, it's not produced directly from the substances we're using as fuels. We've got a lot of steps to go through, as you can see it coming. But we'll do them sort of one bit at a time. The other bit that I've got in here, I'm just going to skim through it because I'm not going to use it in any exam questions or anything. This idea of respiratory quotient. I'm not doing any notes on it, but you know, just for the sake of completeness, it's there. You can measure oxygen uptake and carbon dioxide production in certain living organisms. It's not always easy, but you can do it more easily in animals than you can in plants, because of course in plants you get photosynthesis interfering, because photosynthesis of course uses the carbon dioxide and produces oxygen. So yes, you have problems with that one. But with animals, and perhaps I've put here germinating seeds, you can use a thing called a respirometer, which will give you a reading of oxygen use, carbon dioxide production. And if you do a comparison of those in terms of the, well, you could do it in volumes, you could do it in molar quantities from a balanced chemical equation, you can actually work out what substrate or what fuel is being used, because we have options. The most obvious one, and the one that we're going to trace through this afternoon in terms of the pathways, is glucose. We say, oh, glucose is a source of energy, and we think that's the primary source of energy for living cells. It isn't for a lot of them. Nerve cells and definitely the brain, glucose is all they'll use. Muscle cells actually prefer to use fatty acids. They are much happier with fats than glucose. And if necessary, we can use proteins, we can use amino acids as well. All of those can be fed into the respiratory pathway at different points. And you can work out what's being used by cells if you make a comparison of the carbon dioxide production against the oxygen use. And so you have this idea of the respiratory quotient, which is carbon dioxide evolved divided by oxygen consumed. And like I say, that can come from a balanced equation or just from measurements that you just make experimentally. If you have an organism that's just using, let's say, glucose, you actually find that the amount of carbon dioxide produced in terms of molar quantities is the same as the oxygen used. However, with the other sorts of substrates, fatty acids and amino acids, it's not the same. So if we look over the page, there's some examples. For glucose, you've got the equation that we always summarise respiration into. Hopefully we're going to build that up. But C6H12O6, which is glucose, and other things, of course, plus 6O2 gives us 6CO2, 6H2O, and energy. Well, we've got 6CO2 produced using 6O2, and that's the respiratory quotient, just 6/6, which is 1. So the respiratory quotient for glucose is 1. And so we know that if we're getting a 1-to-1 ratio between the carbon dioxide and oxygen, then that's what's being used. But, example there for a fat called tripalmitin, has a rather wonderful equation for its respiration there. 2 C51H96O6. All right. But if you bear in mind, what do we know about fats? Well, it's glycerol and 3 fatty acids, isn't it? The glycerol will take up 3 of those carbons, leaving 48. The 3 fatty acids will then take up 16 each, I think. That would make sense, wouldn't it? So that's where that goes. But it takes 145 oxygen molecules, or moles of oxygen, to respire that, and we get 102 carbon dioxide on the other side, apart from the water, which we're not concerned with. So if you were to divide the carbon dioxide by the oxygen there, you've got 102 over 145, which is approximately, it is about 0.7, yeah. So yeah, if you cancel it down, 10 over 14, you've got 0.7. So it's a different RQ for fat. So that's all that bit's supposed to be showing you, that you can actually work out what's being respired. And of course, the next question, why is the normal average RQ for humans between 0.7 and 1? We eat a mixed diet, a bit of everything. Okay, let's make a start on this then. This equation is horrifically oversimplified. It doesn't tell us anything about what's going on along the way, but of course, you know even from that first assignment that it's complicated. And we don't look at any real detail at all, to be honest. We're going to condense it down into a very, very simplistic sort of overview. But what we've got to try and do is to trace where all the bits of ATP are going to come from. Now, if you extract all the energy that's possible out of a single molecule of glucose, you can potentially get 38 molecules of ATP. All right, that's quite useful. It's a lot more usable like that. If you were to release all the energy from a glucose molecule by burning it, you just get this big sort of flash. But 38 ATPs, it's like the small change sort of variety of money, rather than a £20 note, 20 single coins, a lot more useful. So what we've got to do is to oxidize our substrate. I've described it as there. Our substrate being our fuel. And the energy comes largely from hydrogen, to be honest. The way that the hydrogen is attached to the glucose molecule, or if we're putting fats into the system, again, it's the hydrogens that are going to be useful to us. So we're going to chase those about for a bit. The other thing that I've put in here is about polysaccharides like starch or glycogen. Obviously, those can be broken down into glucose or any other monosaccharide, to be honest. And if we are going to use other substances, well, they'll just have to make their own way into the pathway because we're not looking at them. All right. So we're going to focus on glucose, and we're going to do this one step at a time. Now, the way I've drawn diagrams and things in here is it's very simplistic, because I probably did these notes along with all the other ones that I keep referring back to when I typed all the A-level notes up. It's getting on for 20 years ago. So yeah, word processors weren't quite as sophisticated as they are now. And there's one or two things I want to modify as we go through, but let's do this step by step. Well, what I put here, this respiration bit is general to start with, and what I should say is we've got the aerobic pathway and the anaerobic pathway. Now, some parts are common to both, and the first step is. So, the first bit, glycolysis. All right, this one's common to both aerobic and anaerobic. So I will put that up here. It's what happens after glycolysis that's different. And this happens, well, whereabouts in the cell does this happen? Cytoplasm, that's it. And it's only if there is oxygen available that the end product of this then goes into the mitochondria. So it happens in the cytoplasm, doesn't use any oxygen. Right, okay. Now, there's all sorts of ways, potentially, of writing this out. If you were to look at it in detail, there are actually about 10 steps to it. We're not going to look at 10 steps because we don't need to. But if you've got any intention of studying anything biological beyond this, you will. And you'll have to draw them all out. And you'll have to be able to name them. And you'll have to describe all the reactions in between. I have now completely forgotten that. So, you know. But what is important to us, really, is what goes in at the beginning and what comes out at the end and along the way. The actual names of the substances in this pathway are not important. But there are two stages, really, to glycolysis. And the first step might seem a little strange, but you'll see how it works. And it's a bit like if you're playing the slot machines or gambling in any way, you have to put some money in order to get more out, hopefully. Yeah? And this is the principle, really, in this first part of glycolysis, that we actually, the first thing we do is use some ATP. But if you think about, you know, what we know in chemistry so far, it's almost like having to get over the activation energy. It's not quite the same, but you sort of have to activate the molecule so it becomes more reactive. So if you have a look at the, I'm going to put a lot of diagram up here, really. The first step is that we start with a 6-carbon sugar, and using up some ATP, we end up with a phosphorylated 6-carbon sugar. Now, it works, it happens in stages. First you get one phosphate group added, then you get 2 phosphate groups added. There's a conversion to fructose along the way as well, but we're not fussed. You know, the names don't matter. So we start off our 6-carbon sugar, all right, for the sake of this example, you know, we are saying this will be glucose. The first step then is it becomes phosphorylated. This is pretty much the same as you've got in the notes. Whoops. Phosphorylated. So nothing's broken down yet, but we have used 2 ATP molecules up to do this. Like I said, first of all, it gets 1 phosphate, then it gets a second. So we're actually going to need to use up 2 ATP molecules. It makes sense if I put the 2 in there, wouldn't it? Because I need to keep track of them. We get 2 ADPs out as a result, plus the phosphates, of course, but we can't add everything in here. So it might have been sensible to colour code this. But that's the first step. We've still got our 6 carbons, so nothing has happened yet in terms of the, well, given that the name is glycolysis, the glyco bit is sugar, lysis means splitting. We haven't done that splitting bit yet. So the next step is that it does get split. We have a stage where there are 2 3-carbon sugars, trioses. They'll actually be triose phosphates at this point. I have just put 2 3-carbon sugar here. They will still have the phosphate on them at that point. And then it's the next step where we start to get something useful, because from here onwards, we get, well, we don't lose any carbons. We've still got 2 3-carbon molecules. And we can now name it pyruvate. And that should ring a bell. Pyruvate, or occasionally it'll be called pyruvic acid. Right, it's unlikely to be the actual acid. But how do we get from there to there? Well, for one thing, we're going to remove those phosphates that we put in at the beginning, and we're also going to remove a couple more as well. And we do that by putting in 4 ADPs, and we get out 4 ATPs. So that's the first thing. In terms of producing ATP, we've now got a net gain of 2. Yeah. So it's like we've put 2 quid into the slot machine, we've got 4 quid out, we are winning. All right, but it cost us 2 quid to get there. Okay. However, you can see that something else has happened. And this is where I need to make a bit of a modification, and I really need to retype this bit, I think, on here, because I've used a sort of old style of representing this. But this is where we meet NAD. Now, NAD got mentioned, when we're talking about enzymes, we're talking about the processing of alcohol. But if you look at the bottom of the page prior to the diagram, you'll see that 4 hydrogen atoms are also released in the second step, and these are picked up by a coenzyme hydrogen acceptor, NAD, nicotinamide adenine dinucleotide, which is reduced to NADH2. Now, that is not how we should write it anymore, all right, but I'll come back to that in a minute. This passes into the electron transport system, where it's used to generate ATP. Aha! So, this is the only ATP we get directly from glycolysis, but I'm going to use a different colour here. 2 of these things, and I'll sidetrack onto them in a minute, go in. They actually only pick up a single hydrogen each, but there's another hydrogen ion that hangs around with them. They actually do extract 2 hydrogens from each of the sugars, but what I'm going to put in is 2 NADH plus H plus, and that's more accurate than NADH2. NADH2 is a bit sloppy because the NAD doesn't actually pick up both of the hydrogens. It only picks up one on its own, but it does have another one hanging around with it. If you want to look at that in more detail, you're welcome. All right, but that's a better way of writing it than NADH2. So I need to tell you a little bit about NADH2, or NAD. Nicotinamide adenine dinucleotide, this stuff here, is made from something in the body. Well, it's known by 2 names. Sometimes it's called niacin, sometimes it's called nicotinic acid. Does anyone recognise having seen those on anything or anywhere? B vitamins, yeah. Yeah. All right, that is niacin, or nicotinic acid, is actually vitamin B3. So, if you are short of energy, and somebody says, oh, try taking some B vitamins, now you know why. Because it helps to make this thing, the hydrogen carrier, hydrogen acceptor, whatever you want to call it, which is really critical in actually carrying the hydrogen to the electron transport system to release energy. All right, and there's another one coming later on. But it's Yeah, I can't think what it's found in, to be honest, but yeah, B vitamins generally are good for energy release, all right, because they literally contribute to this carrier system in the respiratory pathway. So it's very often the way with vitamins that they do that sort of thing, that they are carriers, or they work alongside enzymes. I mean, this is called a coenzyme. That doesn't mean it's an enzyme. It means it works with an enzyme and it helps to carry something for use by an enzyme. What it's carrying is hydrogen. All right, that hydrogen is then going to be processed. In fact, I mean, it's both of those two hydrogens that we've got there, that one and that one, are both going to be processed through the electron transport system to extract energy. So if you're short of vitamin B3, then you're not going to make enough NAD, so you're not going to be able to carry these efficiently, so you're not going to extract as much energy from nutrients as you should be able to. All right? So, you know, when people say, oh, you need plenty of vitamins, well, that's why. You know, they're not just there for decoration. You know, they do have jobs. So, yes, at the moment then, how far have we got? Well, we've managed to break our glucose, our 6-carbon molecule, into 2 3-carbon molecules. And we haven't lost any of those carbons at all yet. And so we're not producing any carbon dioxide as waste at this point. So we've just literally broken the glucose in half. But in doing so, we've managed to get a net production of 2 ATP. But we've also got 2 pairs of hydrogen atoms, if you like. Each one of these NADH plus H pluses is 2 hydrogen atoms in some sort of arrangement. And those can be used later on. If we've got oxygen in the system, they can be used. If we haven't, then they can't, and it's a waste of time. So if you look at the top of that page with the diagram, you can see I've got a summary there for glycolysis. That you start with C6H12O6, and it produces 2 C3H4O3. I'd be hard pushed to actually draw the structure of that, I must say, but that's pyruvate or pyruvic acid, I suspect. But yeah, I have no idea what the actual structure is. 4 hydrogen atoms, right, but those have been picked up by the 2 NAD molecules. And then 2 ATP, which is our net gain, having put 2 in and got 4 out. All right, are we okay so far? Yeah, it's easy so far, isn't it? It does get more complicated. It's trying to remember it all that's the worst bit. We have the poster over on the side for cellular respiration over there, which is quite good in that it uses little dots to represent all the individual carbon atoms. I'll come back to that when we've actually gone all the way through it, and we'll have a look. But I'll also show you a whole load of diagrams that break it down in different ways, because it is, it's something that is not that difficult as long as you get the hang of it at the right time. So we've made our 6-carbon sugar into 2 3-carbon pyruvate molecules. Now, the next thing to consider is everything from here on will happen twice, because each of those pyruvate molecules that we produce there, we've got 2 of them, but each one of them will have the same fate. And we're not going to draw it out twice. So everything from here onwards, you can assume happens twice. And the next stage, as we normally state, would be, oh, Krebs cycle comes next. No, actually it's not. The next thing that happens is something called the link reaction. And it always rather irritates me that the link reaction isn't considered necessarily sort of in its own right. That we just say, oh, glycolysis, Krebs cycle, and the electron transport. And it's never clear whether the link reaction is the end of glycolysis or the beginning of the Krebs cycle. But it is actually, really, I suppose, a 4th step. But it doesn't get its own consideration very often. Now, pyruvate then has got 3 carbon atoms in it. Okay, that's fine, but we actually don't need all of those, and gradually we're going to get rid of them. We are going to completely break down the glucose molecule we started with. And so the link reaction, I think I'll put it, I'm not sure when to start a new board, I think I'll leave that on here. The link reaction takes the 3-carbon pyruvate, and if there is oxygen, it goes through the link reaction. If there isn't any oxygen, then it doesn't. So actually, yeah, I might put that on here and then start a new board. So if we've got no oxygen, you get the anaerobic pathway. If there is oxygen, it then goes to the link reaction. So I'll put that on the next board, I think. And that would occur in the mitochondrion. All right, so I think that justifies a new board. I'm going to put link reaction and Krebs together. All right, these are aerobic only. And it's all determined by the presence of oxygen. So if oxygen is present, pyruvate enters mitochondrion. Okay, so just one of them then. We'll take a single 3-carbon pyruvate. That 3-carbon pyruvate is too big to enter Krebs cycle. And so one of the carbons is lost. And it's lost in the form of what do you think? CO2, yeah, carbon dioxide is the obvious loss. So where's the oxygen coming from to make CO2? Your first thought would be it comes from the air, yeah? It doesn't. Right? The oxygen that we take in from the air is actually used right at the very end of the electron transport system. The oxygen which appears in CO2 comes from water. There's actually a breakdown of water, I think it's the water route. So it's not actually the oxygen, you know, we're not actually exchanging the oxygen that we breathe in for carbon dioxide coming out with the same oxygen in it. All right, so this is different oxygen. And how do we know this? How could you work this out? Radioactive labeling? Come across that idea? I can't think whether we've used it anyway. If you radioactively label oxygen that is breathed in, that radioactive label does not come out in the carbon dioxide that's breathed out. Okay, it actually comes out in water. So water gets ignored a lot, but water is actually a very important reactant, all sorts of places. So from a single pyruvate, we lose a carbon in the form of carbon dioxide. So that's our first bit of waste, if you like. We also get another NAD comes along. And the NAD picks up another pair of hydrogen atoms. I was putting those in over this side, wasn't I? Were they coming in red, I seem to remember. So I've got an NAD coming in here, picks up another 2 hydrogens, and we get, I'm going to insist on writing it properly. All right, so we get another one of those. Of course, the other pyruvate's going to be treated exactly the same way. So at this point, we've then got a 2-carbon piece left over. All right, we had 3 carbons, we're now down to 2. The old name for a 2-carbon group was acetyl. Yeah. Of course, what would we now call it? Ethyl? Yeah, think back to the organic chemistry, that it's eth if it's 2 carbons. So ethyl and acetyl mean the same thing. But we're doing this with biology hats on, so we still call it acetyl. All right, and it's one of those things that, you know, I don't know whether biologists do it on purpose just to annoy chemists, but they've changed some of the names and they've not changed others. And I can imagine that one of the worst degrees to go into would be biochemistry, because you've got this complete contradiction between the two, getting the names. I think you have to be more of a chemist actually to do biochemistry. It's not something I'd want to do ever. But we're left with this 2-carbon group, and we're just vaguely going to call it an acetyl group. So it's a 2-carbon acetyl. I will put in brackets ethyl, just as a gesture to the chemists, because I sometimes pretend to be one. So a 2-carbon acetyl group. All right, that'd be C2H5, waiting for something else. So a 2-carbon acetyl group is left if we take our carbon off here. Now, this is going to go into Krebs cycle. I should say the Krebs cycle. Otherwise, it would need an apostrophe somewhere, and it doesn't have one. It's the Krebs cycle. I'm trying to work this one out for years. But it has to be carried into the Krebs cycle by another carrier. It's another coenzyme molecule. And it's called coenzyme A. I'm guessing this was one of the first coenzymes to be discovered. It doesn't have any fancy name. It's just called coenzyme A. And so what coenzyme A does, it comes in here. Oh, let's have another colour for that. Well, my coenzymes seem to be red, so oh no, let's stick another one in. Coenzyme A can be green. All right, that comes in and it picks up these 2-carbon groups, and we end up with something you'll see described as acetyl coenzyme A. Sometimes that's abbreviated just to acetyl CoA. Coenzyme A has got no actual role in the respiratory pathway apart from this. It's just a carrier. All right, so it's like a shopping trolley, if you like. It comes along, picks up these acetyl groups, and it's going to dump them into the Krebs cycle. All right, the coenzyme A is not changed in any way. It is literally a carrier molecule, like the NAD up there is carrying hydrogen. Coenzyme A comes along and it picks up these 2-carbon groups, and it's going to dump the acetyl group into Krebs cycle, and then it leaves it and goes and gets some more. Right, so that's the link reaction, and that's all they call it, the link reaction. You know, like it's just nothing's happening. It's incredibly important. There's a loss of carbon dioxide. There's a production of reduced NAD, the NADH plus H plus. We usually refer to as reduced NAD. Actually, I might go back a slide to where I first introduced that and just mention it under there. So NADH plus H plus equals reduced NAD. Sometimes how you'll see it written. I'm just trying to cover all the possibilities because I'm aware that it's written in a lot of different ways. Yeah, so you've come across the reduced NAD before. So yeah, we've got different ways of writing that. You could put NADH2, that's not very accurate. The NADH plus H plus tells you exactly what's there, but reduced NAD is it's a bit of a get out because it doesn't tell you anything about the number of hydrogens it's carrying, but it's a slightly easier way of saying it, I think. So we've made another one here. All right, so you got another one of those there. You've got the loss of the carbon dioxide over on the other side over there. So we've actually got a lot of significant stuff happening. So this is the link reaction. So the next step then is the Krebs cycle. So I need a bit more space for this. Now, Krebs cycle is, as it implies, a repeating sequence of reactions. And this is about another 10 conversions between different substances. The only detail I've given you in the notes is the names of a 4-carbon substance that picks up the 2 carbons here, and then the name of the 6-carbon substance that's produced. And what we have, it doesn't matter which direction we write it particularly, is a 4-carbon substance called oxaloacetate. Again, you can see you've got acetate instead of ethanoate. This is the problem. Biological names are so complicated anyway. All right, so that's going to come around, pick that up. Right, that got fed in here, didn't quite show that. And it makes a 6-carbon substance, 6-carbon citrate. Okay. We then have to get back to the oxaloacetate for this to be a cycle. And in doing so, we've got to lose those 2 carbons that have just been put in from the acetyl group. And you can find out where they're going to be lost by radioactively labeling them if you're interested in that sort of thing. And yeah, people have spent many, many years sorting that out in order to work out what's happening here. But this sequence of changes between citrate and oxaloacetate, again, if you study biology beyond this, this is where you'll have to be able to draw all the intermediate substances, name them, explain the reactions that are going on, work out what enzymes are involved. And we used to, I can remember when I started doing A-level biology at school, we had to try out the classes before we committed to them. And I can remember when I just finished what's now year 11, we took O-levels then because it's GCSEs now, but once we'd finished our exams, we had the sort of sixth form taster week, if you like. And I remember going, even now, and this is a long time ago, we're talking over 40 years ago, we went to these classes in the different subjects, and I can remember really clearly going to the biology taster session, and they took us through this and made us draw out all the chemical formulae and the names and all that. They were trying to get rid of us. There were too many people who had chosen to do biology, so they were really sort of trying to put people off by getting them to draw all this, but I thought that was fascinating, so I stuck with it. And yeah, then made a mess of it when it got to the end. But the sequence, we used to have to learn them, but then I think somebody has sensibly decided that actually it's not what happens in between that's important, but it's the inputs and outputs that matter. But my husband, in his year, I don't know whether it's a standard mnemonic for remembering all the intermediate substances, but they have a way of remembering all the intermediate substances. And so it goes citrate, alpha-ketoglutarate, succinate, fumarate, malate, oxaloacetate. See, I can get them right. The fact that there's a fumarate in there that begins with an F gives you a rough idea of what the mnemonic might include. But those are the main intermediates, and then there are others as well. And they all are slightly different from the previous, and sometimes it might look like they're getting bigger before they get smaller. But what you have here is a sequence of what we would describe as oxidation and reduction reactions. And that's what respiration is all about, oxidation and reduction. Now, our final learning outcome in chemistry is redox, which is reduction and oxidation. But it's very, very isolated in the chemical sense that you don't necessarily see the use for it there so much, apart from the fact it can determine whether a reaction will happen or not. But with biology, respiration and photosynthesis are entirely oxidation and reduction going along sort of side by side. As soon as, well, as one thing is oxidized, another thing must be reduced and vice versa. So very, very complex, but yeah, a lot of big chemistry in it. The way that we then get back to oxaloacetate, so this thing then is the Krebs cycle. All right, a cycle implies that it starts in one point and then circles back to that. You'll see that the way I've drawn it has got carbon dioxide coming off in different places, ATP and hydrogen coming off in different places as well. I'm just going to summarize those bits actually because it doesn't really matter where they come off, but we get along the way between the 6 and the 4, which takes several steps, we must lose the other 2 carbons that came in up here. We're going to lose them. So you get 2 carbon dioxide molecules produced. Remember, this is just for each pyruvate. So for each glucose, this will happen twice. So we've now got rid of all the carbon dioxide. We also get just a single ATP. Oops. 1 ATP. So it's not very productive in terms of ATP. We've not actually managed to make much ATP yet, have we? You know, for a single glucose, so far then, we got 2 out of glycolysis, and we get 2 turns of this for another 2. So all we've managed to make is 4. And yet I said you can get 38. So the other 34 are going to come out of the electron transport system. All right, now the other thing that's happened here, we get, let's do this the right way round, 3 NADs come in, and we get 3 of those reduced NADs, so NADH and H pluses. So a pair of hydrogens picked up by each of those. Sorry, that little dot came from nowhere. But we also get another carrier, another coenzyme hydrogen acceptor. This one's called FAD, and you only get 1 of them come in. And that picks up 2 hydrogens, but this time they do both attach to the FAD, so we can actually write that. Now, I need to explain what this one is. This one, oh, I don't want an arrow on it. That implies it's going somewhere. Right. This one is called flavine adenine dinucleotide. And this one comes from, if you've ever read your cornflake packets, you might have seen these. But this one's a vitamin called riboflavin. Sorry? All right, riboflavin. All right, so riboflavin is another B vitamin, and this one is B2. And it's only today that I found a way of remembering which of those was which. Every year before I teach this, I have to look them up because I forget which B vitamins they are. But what I do remember is how their hydrogens are processed in the electron transport system. Weirdly, I know that the hydrogen that comes off the reduced NAD generates 3 ATPs each time it's processed. So I now know that's vitamin B3. And the hydrogen that comes off the FADH2 actually only generates 2 ATP. So I now remember that's a bit of vitamin B2. I've actually, by knowing something else, I've got a way of remembering which B vitamin it is. But yeah, it's a sort of reverse sort of memory thing. All right, so what have we got then? Well, we've produced quite a lot of stuff, but not an awful lot of ATP. And so if you have a look just under the diagram for the Krebs cycle, I've tried to summarize everything to this point. So the overall reaction for glycolysis, acetyl-CoA production, that's the link reaction, and the Krebs cycle, you could summarize it as follows. C6H12O6 plus 6H2O. Now, they haven't appeared anywhere in the processing that I've drawn up because they're not, I don't know, they're not evident, I suppose, until you start looking at individual reactions. Produces 6CO2. All right, that's fine because we produced 2 here twice and then 2 in the link reaction twice. So there's our 6CO2. 4 ATPs, we've just found those. All right, twice around this will produce 2 and 2 in glycolysis, 4 of those. But we also have 12H2 in the form of the reduced coenzymes, and you can track them all down. They're the things that are going to be really useful. All right, so all we've got so far, 4 ATPs and then 12 pairs of hydrogen atoms, if you like. So let's just track those 12 down. This is where I have to cross my fingers and hope I've got them all. Right. We have 2 pairs here, right? So there's 2 NADH and H+. That's like saying 2 lots of NADH2. So we've got 2 H2s, if you like, there. No problem. Okay, link reaction happens twice, so we get another 2 there. All right, 1 for each pyruvate. So we're going to get 2 lots of that, and so we've hit 4. We've then got 2 lots of what happens in here. And we've got 2 hydrogens being picked up by each of 3 NADs. So that's, hang on, let's see if we add those to the one pair that we've got picked up by the FAD. There's 4 pairs for each turn of Krebs cycle, so that's 8. Add those to the 4, we've got our 12 pairs of hydrogen atoms. See why I need to do this all in one go. So that sort of makes sense. The only thing we haven't really accounted for was the water. Don't worry about it because it's going to disappear. Okay, that leaves us with the electron transport system. Now this has other names as well. Sometimes pathway, sometimes just respiratory chain. Another option. Oops. Oh, sorry, that was a bit squiggly. Okay, now what we've got to process here then, the function of this and where it's located, this is what the internal surface of the crystal is for. All right, so this, what we didn't put actually, Krebs cycle, let's just go back to that. I didn't even label it in there, did I? So Krebs cycle. And that happens where? Yes, mitochondrial matrix. Okay. Now, yes, now is when we need our huge surface area, the infolded membranes of the cristae, and you have this arrangement of carrier molecules, channels, if you like, on the inner surface of the cristae, which move hydrogen ions and electrons between the matrix and what they call the intermembrane space. So, let's see if I can draw this. Probably won't be just a section of it. Right, there's the outer surface of the mitochondrion. Oh, let's just do a whole one. It's a bit easier. Let's see what's what. You've then got this arrangement. Well, I'm not going to draw a whole one here, just like that. And we're looking at a process across here. Whoops, that's supposed to be a little arrow. Right, and we have, what's going to happen is hydrogen ions are going to move that way, and then we get electrons moving that way. No, we get movement in both directions. I don't want to overcomplicate it, but we get this movement across that membrane. All right, so we've got the outer membrane. Ooh, inner membrane. And then between them, you have the intermembrane space. So, this huge surface area is going to give us a lot of scope for generating energy. And we have a sequence of carrier molecules called cytochromes associated with an enzyme called ATPase. Aha, there it is. I'm not going to try and draw those as they are often depicted, because you don't really need them like that. What I'm going to draw them as is really just to sort of try and explain the nature of this process. It's very often drawn, try to think how best to put this, as though you've got arrows where one substance is oxidized, another substance is reduced, and then as that one's reduced, that'll oxidize another one. Then when that one's oxidized, something else is reduced. So you get these two-way arrows going along this sequence of carriers. But the effect in terms of energy transfer is as though you've got like a sequence of steps. Oh, I can never remember how many I need, really. I think that's probably one too many there. It looks like one too many. We talk about 5 of them, but this is just sort of, diagrammatic sort of version of what's happening. And your electron transport is actually a little bit confusing in terms of its name. We call it electron transport because, well, just a reminder, a hydrogen atom equals a proton and an electron, right? Now, what's actually traveling here are hydrogen ions, in other words, protons, and their associated electrons. So if we talk about proton pumps and things like that, and movement of protons, we're talking about hydrogen ions, but the hydrogen ions have these electrons associated with them, so we call it the electron transport system. All right, it's sort of, if you look in the depths of the biochemistry, it makes more sense, I guess. But what's going to happen is that our NAD, our reduced NAD, so this comes in here, it delivers its 2 hydrogens and then goes off to get some more. All right, so the NAD is freed up at that point, and that is going to be really important when we start thinking about what if there isn't any oxygen. those two hydrogens are then sort of passed between the carriers in this system, and it's rather as though you have a ball bouncing down some stairs. At the top of the stairs, it's got a lot of potential energy, yeah? And with each bounce down the stairs, it's losing potential energy until when it gets to the bottom, it has none. And so what we have, the effect of this is that the hydrogen is passed between the carriers, and what we end up with at the end of it is still hydrogen, but it's got much less energy. And at that point, I suppose here we should really say this has been the hydrogen ion and its electron being passed between the carriers, and then they rejoin when they get to the end of the chain. And the energy is really extracted from the electron, and that is often the way. Electrons, of course, can be at various different energy levels, and the energy is extracted to make the ATP. And if we put in the hydrogen from our NAD, our reduced NAD, you have enough energy released to make 3 ATP molecules. That's what I was saying is helping me to remember now which vitamin they come from. That's the sort of effect that we get. It's very much not how it'll be represented in diagrams, but I picked up a whole load of diagrams this morning that I thought might help me. I'm going to go over this afterwards. The other source, of course, was the FAD, and I can't remember what colour I did that in. Hang on, was that blue? It was blue, yes. Right, okay. Let's put the FAD in here. The FAD brings in its 2 hydrogens at this point. All right, so they come in lower down. And for each one of those, you only get 2 ATP molecules. So we've got to do a bit of tallying up when we get to the end of this, because, you know, we've got these from all over the place. How many of them have we got? Will it add up to 38? Who knows? It's always a bit hit and miss. I do have years where it undershoots and overshoots, so we'll see. But there's something left at the end here. What we've got at the end of the chain there, when we've extracted the energy from its electrons, we've now got low energy hydrogen. You can't just leave hydrogen hanging around, can you? Easy way to mop up hydrogen is turn it into water. So we will need oxygen, and that's where the oxygen comes in, and I've run out of colours. So let's have a yellow. Well, that's not going to stand out very... Oh. Sorry. It doesn't want to go away. Right. The purpley colour should we have? Right, oxygen comes along, picks up the hydrogen, and I know this doesn't balance, but that's where you get the water. And that is the only place in respiration where oxygen is used. All right, the oxygen that our lungs take out of the air, picked up by our red blood cells, delivered into the tissues, is used at that point and that point only. All right, nowhere else along the way. Okay, let's do a bit of tallying up here. See if this actually adds up okay. Right, so where does all our ATP come from? So ATP tally. Glycolysis. What did we get out of glycolysis? Well, we did get 2, didn't we? We got 2, well, let's put the direct ones, and then let's put other as the sort of, so they're not going to be added up yet. So we got 2 ATP, but we also got a couple of reduced NADs. Yeah? So they're not counting yet. When we did the link reaction, what did we get there? Well, we got 1, didn't we, for a single pyruvate. So it's actually going to be 2 reduced NADs. All right, so that's times 2, remember. Krebs cycle. Again, times 2. And what do we get out of that? We did get, yeah, so there's 2 circuits, so we get 2 ATPs, plus we got 2 lots of 3, didn't we? So we've got 6 of those. That's right, and 2 of the reduced FADs. Right, okay, so that leaves us with electron transport system. Right. We've got, well, how many reduced NADs have we got to deal with? Should be 10 there. All right, so you've got 6, 7, 8, 9, 10. So 10 AD, that's it. So those will give us 30. And then the 2 of those will give us, they only produce 2 each, so 4, yeah. And if we're really lucky, that lot adds up to, should be 38, yes. And there we go, that's where they all come from. Did I turn that on? Yes, I did. Right, just to add on to here then, that the use of oxygen and the production of ATP by phosphorylation means that, actually let's write it in purple. So oxidative phosphorylation is the process that occurs in the respiratory chain or electron transport system. So oxidative phosphorylation. Just before I divert and show you some diagrams to look at this in other ways, what if our substrate wasn't glucose? You know, this is all very well. So, oh, we put 6 carbons in at the top, break it down into 2 lots of 3 carbons, then we lose 1 in the link reaction, then lose the other 2 in the Krebs cycle. But not everything starts with 6 carbons. Well, you've got a lot of points in this pathway where you can feed other types of compound in. And if you turn over to, you've got a couple of diagrams, several diagrams, but opposite there's a answer sheet for some dominoes, you'll see a diagram that sort of gives you an idea of where other substances can be fed into the respiratory pathway, or indeed where you could actually take substances out. And if you look, for example, well, you know, you go down that middle bit, you've got the standard carbohydrate, you might have glycogen in storage, you can convert that into glucose, but glucose back into glycogen, and the glucose is then fed in through the 3-carbon sugar to pyruvic acid and so on. But you could, let's say, take the proteins made of their amino acids, and amino acids can generate molecules that could be fed in at just about anywhere, to be honest. You can have amino acids contributing 3-carbon groups that could go in through the triose, the 3-carbon bit of the first part, the glycolysis section. Similarly into the pyruvate bit, or you could have amino acids giving you 2 carbon groups that could go into the acetyl-CoA bit. Or you could use amino acids to generate molecules that go into the, well, they've called it citric acid cycle for the obvious reason that one of the substances there is citrate or citric acid. And in the section that you've got in your notes on enzymes, we had one type of enzyme was called a transferase, and it was transferring amino groups from one type of acid to another, and it might have been generating new amino acids, but it can also turn amino acids into what are essentially Krebs cycle acids. So if you wanted to use protein for energy, that's all you have to do. You break it down into amino acids and then take the amino group off and stick that somewhere else, or get rid of it in urea, and then what's left of that amino acid can be usually fed in as a Krebs cycle acid. So, you know, they're very, very usable as respiratory substrates. Over on the other side, you've got the fats. The triglycerides are very useful for energy. And those are broken down into glycerol and fatty acids. We're familiar with that. And glycerol, that 3-carbon alcohol, not surprisingly, can be fed into the 3-carbon sugar bit, the triose phosphate bit. And the fatty acids have some very specific processing pathways. Where it says beta oxidation of fatty acids is sort of one of the standard biochemical pathways, where what happens if you take those fatty acids with, let's say, 16 carbon atoms in their great big long hydrocarbon chains, you just chop them up into 2 and then 2 and then 2, and they sort of cycle round. So you chop 2 off, feed that in, chop another 2 off, feed that in, chop another 2 off, feed that in. And of course those 2 carbon groups, what we would call ethyl groups, could also be called acetyl groups. So they can go straight in acetyl-CoA. So fats provide an immense amount of energy, as we well know. Because, you know, if you've got a single fat molecule with 3 fatty acids, if they all had 16 carbons in them, right, that's 8 pairs of carbons in each one. So that's 24 pairs, you know, 24 acetyl groups that you can put into respiration. So it's not surprising that, you know, there's a lot of energy to be had from fats. So they're very, very easily fed in. So, you know, this idea that, respiration, this pathway only applies to glucose, you know, is very, very narrow. And you do have to bear in mind that a lot of the other substances that we've met can go in via a variety of different routes. And, yeah, there's very little that's not fair game as a respiratory substrate. The only thing that's not on there that we have met, of course, is nucleic acids, but you could break those down, couldn't you? You know, you can break down, adenosine, for example, or, you know, you've got the ribose in there, or deoxyribose. Well, what do you know? It's a sugar. We can stick that in. I don't know about the bases, to be honest. I think they're probably too much use in the body that I don't think we'd use those for energy. And the phosphates are forever recyclable, aren't they? So, you know, anything can be broken down. Right, what I did this morning while I was drinking my tea and eating chocolate cake for some strange... I had this urge to make chocolate cake yesterday and it went horribly wrong. I don't do cakes. It ended up with me eating cake for breakfast. While I was doing that, I was just thinking I could do with a few more diagrams, and then I couldn't decide which ones I wanted. So I thought, oh, well, I'll just put a selection on and we can pick and choose from what we like. Now, I rather liked this one. This is my first choice. I thought, oh, we'll leave it at that. And then I found another and another and another, and that's how it goes. But as a really nice overview, except that I've got a slight... Oh, no, that's not right, is it? What's wrong? Yeah, 36 ATP off the electron transport system. What should we have? We should have 34, but where have we got the other 2 from? Yeah, we should have them... Oh, I'll have to get rid of that, won't I? So that should be 34. And we should have 2 coming off here. All right, so there should be another 2 ATP. 2 ATPs should have come off there. Yeah, I know, that should have been a little yellow burst of whatever. That's the trouble. See, I wasn't concentrating, but it looked neat. And when you're trying to revise it, you're trying to see the whole picture. Diagrams like that are really helpful. There is one in your handouts, but of course they're in black and white. So if I were you, I would try and hunt down a nice coloured one, but not this one. Yeah, there are some good ones about, and I think that's probably the one that most people see when they are looking for diagrams. And actually, it's really quite a decent one. It doesn't give you any quantities though. So you've got your glycolysis over here on the left, producing pyruvate. Some of the electrons carried via NADH up at the top there, it just means the electrons as part of a hydrogen atom, carried off to the electron transport chain over on the right there. Pyruvate goes into the mitochondrion. We haven't got a link reaction here, so that's missing. And this is it. Trying to find the perfect diagram is difficult. Krebs cycle is giving us a little bit of ATP and some electrons. And then most of the electrons coming from the end process there. ATP synthase or ATPase, same thing really. So yeah, no idea of quantities, so yeah. I was getting a bit fed up by this point. This one is a bit more complex, and this one includes the problems that occur when we've not got oxygen, and what are we going to do to get some sort of energy release if there isn't any oxygen, because if there's no oxygen you can't run the electron transport system. If you can't run the electron transport system, you don't free up any NAD, so you actually are at risk of having the whole thing grind to a halt. So I'm going to come back to that when we talk about the anaerobic side of it, which I'd rather do another day because I don't want it to get too confused with this. Sorry, I've gone backwards instead of forwards. I thought I'd sling that one in as a more thorough diagram of the Krebs cycle. And it gives you a little bit more detail, and that came from the Royal Society of Chemistry. Now, they're actually quite good at explaining, sort of chemistry for biologists, or, perhaps biology to chemists. I'm not sure which way around. But this includes the names of those compounds, and the bit that we're really interested in, curiously, their numbering system on here. I didn't go actually to the website and see what the numbers represented, but if you were running this, you would actually be starting at arrow number 2 up there, where oxaloacetate is picking up the 2 carbon groups off acetyl-CoA. So yeah, quite why number 1's over this side, I don't know with that one, but you can see where the reduced NAD is produced, you can see where the carbon dioxide is produced, you can see where the reduced FAD is turning up down the bottom there, and it actually sort of takes you, round the whole lot in quite a simple sort of way. I did quite like that, but it is more than you need. You don't need to know all the intermediate names. Like I say, I only know them because of a mnemonic, which doesn't really help. But yeah, so that just looked useful. And then this, right? Oh, this you need to know. This is well, one of many, and it was just a matter of choosing an example here, really. But this shows you the membrane of the inner membrane of the mitochondrion, the cristae. So you've got your phospholipid bilayer there, and then you've got your electron transport chain represented by these sort of reddish orange coloured blobs, which are the protein channels running through there. And the process is sort of not clear, but almost almost clear, that you've got hydrogen ions going from the lower part of this diagram through these channels up here, which are this is the actual chain across here. All right, you've got 1, 2, 3, 4 obvious molecules with others associated, and hydrogen ions are being pumped across into the intermembrane space, which is identified up at the top there. So we get all these hydrogen ions building up in there, and then they make their way back that's this chemiosmosis, this movement of ions from the intermembrane space back into the matrix via this enzyme labelled here as ATP synthase. ATP synthase is a much more helpful name, I guess. It talks about the making process, doesn't it? Synthesis. And so over on the right there, that great big blue blob is allowing the hydrogen ions to come back through and using the energy to make ATP. So that was sort of quite a useful sort of diagram, that one. I then found one that had got more of, it's got that as part of it. I don't know whether I put that on. Hang on, I'll come back to that. No. This was a small part of a larger diagram, and you know, if you're looking at that and thinking that's horrific, I don't want to know anything about that, that's fine. But actually, if you break it down, it's not as bad as it looks. Right, the other bits that are on here are to do with the anaerobic process, and I'm just trying to avoid that at the moment. So let me just park this. Get rid of that, and then I'll reuse. I might rearrange bits on it and make it available at some point. Right, the point that we've reached then really is that we've gone through the aerobic pathway. I might as well carry on now, I suppose, into the anaerobic pathway because we're still got some time left. It won't take me that long, and then that'll take us completely to the end of the content for this, and then we can spend Wednesday morning having a look at the, there's a set of dominoes to do in here, having a look at the exam questions on here, and I can also give you the revision questions for the exam, and you can start working on those, and that will give us a bit of a head start on next week's work. Right, let's get back to this then, because let's check that is on, isn't it? Yeah. The problem that we've got here then is the oxygen. All right, if the oxygen's available, it's picking up the hydrogen. As long as the hydrogen can be removed at the end, it can be fed in at the top. But if there is no oxygen, then the hydrogen down here, all right, this hydrogen here, is going to remain in this system. And if this gets full of hydrogen, then it can't be released from up here. And if all the NAD is reduced, if all the NAD that a cell contains has got hydrogen attached to it, then you run out of NAD and everything grinds to a halt. So there are options for freeing up a little bit of NAD, and both options are only sufficient to keep glycolysis running as an energy supply. So it's a very, very inefficient sort of respiration, and it takes us right back to the production of pyruvate, because if we get to this point and there is oxygen, then the pyruvate goes into the mitochondrion through the link reaction, Krebs cycle, electron transport keeps running, and we get our full 38 ATPs. On the other hand, if we haven't got enough oxygen, then the pyruvate can't go into the mitochondrion, and you have to use an anaerobic pathway to free up enough NAD just to keep this running. And all you can do is just keep glycolysis going, which of course produces a net 2 ATPs in comparison with 38 for the whole process. So anaerobic respiration is 19 times less efficient, all right, that for each glucose it will only produce 2 ATP rather than 38. And you know, the thought you have in your mind then is, well, that's very wasteful of glucose then, isn't it? Well, no, because it's a partial breakdown, and anaerobic respiration can't carry on for long periods of time. And what tends to happen is that it can be used for short periods, perhaps in human muscles, typically no more than a minute, and that's pushing it somewhat. But you've got recovery time at the end. What you do is build up an oxygen debt, and once you can get the oxygen back into your system, a lot of that oxygen is actually used to reconstruct glucose. And we have these two possible pathways. And so the anaerobic respiration, I think you've got this in the notes. All right, if we've got a lack of oxygen, you will find, yes, we go back to it, yeah, we can't do anything with the pyruvate, and this build-up of coenzyme is a bit of a problem. And the options are, well, lactic acid production is the one that we're familiar with in our muscles. Now, there are some benefits to training, you know, sports training, are that it improves our resistance to lactic acid. But lactic acid being produced by anaerobic respiration is much more common in one type of muscle fibre as opposed to the other. So we have the two types of muscle fibre. I think I've mentioned them before, the red ones and the white ones. Red muscle fibre is red because it contains myoglobin. Remember that from circulation? Myoglobin is an oxygen store, so you don't get the anaerobic process going on in there. The white muscle fibres, on the other hand, don't store oxygen, and they're not very well endowed with mitochondria either, so they are used to working anaerobically. But in order to make them work efficiently, you have to train and actually encourage them to be sort of resistant to lactic acid. The problem that lactic acid causes in the body is muscle soreness. They used to think it caused cramp, but now they don't. I don't know what causes cramp. But it's not lactic acid. And the other thing with lactic acid, of course, is that it disturbs the biochemical balance in the cells, because it's a change in pH, isn't it? It does help the body to unload oxygen from hemoglobin easier, but, you know, we're not really using oxygen here anyway, so, yeah. But lactic acid can be a real issue. And so what we're doing really is we're just taking pyruvic acid. I was just wondering if, no, I haven't got its formula there, but pyruvic acid is not put into the mitochondria, and it doesn't go through the link reaction and Krebs cycle. What it does, well, and I draw this slightly differently from how it is in the notes, that we bring in the reduced NAD that we've managed to create. That's the problem, it takes up too much space. It comes in and it gives its hydrogen back again. And you think to yourself, well, what was the point of that then? Well, it has caused the production of a small amount of ATP on the way. But in doing that, we get lactic acid. All right, so the lactic acid is just a sort of product of pyruvic acid plus some hydrogen that frees up the NAD. This is then going off to glycolysis. So it keeps the glycolysis running, but that's it. You know, we're breaking down a lot of glucose to produce not very much energy. And in doing so, we get this oxygen debt. The other option, if you're not an animal, but you are, well, a fungus, I don't know whether plants do it generally, but I think it's fungi generally. And this quite neatly sort of takes us back to what we were doing with the enzymes. Um, we called it there alcoholic fermentation. There are different sorts of fermentation. I made the stupid mistake of arguing this with my husband, who, because he works in the food industry, and particularly in baking, I was arguing that fermentation is always anaerobic, and he said, no, it isn't. And yeah, well, I had to give in the end. He is actually the specialist in that one. But we use yeast in two different ways. Yeast, when it's used to make bread, of course, it's aerobic, and that's one of the reasons that you need the bread, and you get oxygen into it, and you let it rise, and that's aerobic. It's using oxygen, and it's producing bubbles of gas, carbon dioxide, and then when you cook it, you kill the yeast. But of course, then we use it anaerobically to produce alcohol. But yeah, the debate as to whether fermentation meant it had to be anaerobic, went on for quite a while. And in the end, I couldn't be bothered to pursue it. But the idea in a situation where there's not enough oxygen, that's, yeast will use the pyruvate. And I don't know if any of you have ever made your own beer or wine. You actually have to keep the oxygen out of it. With beer, it's not a problem because that produces a froth on the top, and that keeps the oxygen away from it. If you make wine at home, you have a bottle, a demijohn, with a sort of a trap at the top, an air trap that stops air getting into it. If you let air get in, well, it'll just respire all the glucose away, and you get nothing, really, because it will respire it away aerobically. So you don't really win if you let the oxygen in. So what you get with the alcoholic fermentation, this is exactly what we were looking at last week, that the pyruvic acid on this occasion, first of all, goes to ethanol, all right, which is our 2-carbon aldehyde. And I'm just trying to think whether I think I drew out the formulae for this, didn't I, at some point? So ethanol, that's the nasty stuff, which you then have to try and remove. And I was talking about us doing the reverse of this in our livers, if you remember. And from ethanol, we then have the conversion that involves the hydrogen. All right, so this is where you get the NAD, reduced NAD, sorry, let's write it with enough space this time, coming in, delivering its hydrogen, and going off again to keep glycolysis running, and that's the best that can be done. And that was where we had the enzymes written in there, alcohol dehydrogenase. Okay, and I say that's actually named by the reverse reaction, which is sort of equally valid because that's what the liver does to break down ethanol and produce ethanol. So some people then can't get rid of the ethanol. And at that point, I think I was talking about it turning into ethanoic acid. But this makes a lot more sense, actually turn it into pyruvic acid. Somewhere we must have lost some carbon here, mustn't we? Because pyruvic acid's got 3 carbons, ethanol's got 2, so somewhere in here we must lose a carbon dioxide. No idea quite where. I've missed that out somehow. Yeah, incomplete diagram. But yes, if you're producing alcohol, if you're making your own beer or wine, you get these bubbles. That's that. Right, to get from 3 carbons to 2, we must be losing one somewhere. And whether it's possible for the body to process ethanol back to ethanol and then go to this, to pyruvic acid, I'm not sure, because that would mean adding carbon in. And it's not easily available. You couldn't just take carbon dioxide and use it like that. So sometimes pathways are not immediately, obviously reversible, but that, you know, that's an option. Obviously what we're looking at here is yeast, which is different. Now, let's just get back to that. Pick up the anaerobic bits on here, and hopefully that will all make some sort of sense. Right. I will, yeah, I'll tidy these up somewhat, but this one, you can see, I hope, now we've got, this is a lactic acid one, where we've got our glucose being taken into the cell, goes through glycolysis. If oxygen is present, the pyruvate will go into the mitochondrion. That will generate a whole lot of ATP, carbon dioxide and water. If oxygen's absent, it goes through a fermentation process. We tend not to call it that in mussels, really. But in doing so, the lactate is released, and NAD, described there as NAD+. If you're going to be really picky, that's what it is. NAD is released so that that can go back and keep glycolysis running, and then it goes through the fermentation, releases NAD again, and it is just enough to keep the process of glycolysis going, but nothing else. Doesn't relate to that. Doesn't relate to that. That was an overview. I think I need to rearrange the slides, tidy them up a bit, and then I will put this on Moodle. With this one, if you start at pyruvate, you get the sort of obvious bits. If you've got oxygen present, through acetyl-CoA into Krebs cycle and the rest of it. If there's no oxygen, you've then got the two options, one either side. Over on the right there, without oxygen, you've got the production of ethanol, and there's the carbon dioxide coming off that was missing from my diagram. Again, enough NAD plus, as they describe it, to keep glycolysis going. And similarly over this side, to produce lactate, same thing. So two different pathways, but producing the same outcome, and that is to free up the NAD to keep the glycolysis running. And yeah, that was the last bit I found, which I didn't check properly. It looks like I hadn't checked the others anyway, but that had got the actual chemical formulae in, so that was a little bit clearer if your chemistry was good enough. That with the lactic acid formation on the left here, glucose goes through glycolysis to produce pyruvate, and I like these because you've got this sort of continuous cycle in the middle. That reduced NAD that was made by glycolysis was then processed via the pyruvate to regenerate NAD that could be used by glycolysis to produce the reduced NAD, and so on. And the glycolysis was churning out the ATP at the top quite nicely. And similarly over with the alcohol production over on the right there, same sort of setup, so glucose to pyruvate. The pyruvate conversion to from the pyruvate to, well, that's ethanal. We used to call it acetaldehyde. It's an interesting mixture there, actually, of terminology, but that's ethanal. And converting that into the ethanol uses the hydrogen. So there's an extra step in that side of it, but yeah, I will tidy that up and I'll put that on Moodle. Right, I think we will stop at that point, because that's enough chemistry for biology for one afternoon.